Section 1, the first section of the NIH guidelines, provides the scope of the NIH guidelines. It defines the purpose of the NIH guidelines and recombinant and synthetic nucleic acid molecules, as well as providing general definitions used throughout the document. It identifies the applicability and compliance requirements of the NIH guidelines at institutions. Section 2 of the NIH guidelines identifies and describes the following as they pertain to the research subject to the NIH guidelines: risk assessment, risk groups, and containment requirements. The section that will identify where your research falls in regards to the required approvals necessary to conduct the work is found in Section 3, Experiments covered by the NIH guidelines. Sections 3A, 3B, 3C, 3D, and 3E all outline non-exempt experiments that require IBC review. Section 3F experiments are those recombinant and synthetic nucleic acid experiments that are considered exempt from requiring IBC review. At UWM, these experiments are still required to be reviewed and registered with the Biosafety Program Office prior to commencing or continuing activities. Let's discuss these subsections in greater detail. We will now discuss Section 3 of the NIH Guidelines, which is the section in which the 6 categories of experiments that involve recombinant DNA or synthetic nucleic acid molecules is outlined. Section 3A are the experiments that require NIH director approval and the Institutional Biosafety Committee approval before initiation. These include the deliberate transfer of a drug resistance trait to microorganisms that are not known to acquire the trait naturally, if such acquisition could compromise the ability to control disease agents in humans, veterinary medicine, or agriculture, and they require NIH director approval prior to any kind of initiation. Section 3B are experiments that require the NIH Office of Science Policy and Institutional Biosafety Committee approval before initiation. These include experiments that involve the cloning of toxin molecules with an LD50 of less than 100 nanograms per kilogram of body weight, or experiments that have been previously approved under Section 3A1A as a major action experiment under the NIH guidelines. Section 3C are experiments that involve human gene transfer that must require Institutional Biosafety Committee approval prior to initiation. These involve experiments that involve the deliberate transfer of recombinant or synthetic nucleic acid molecules or DNA or RNA derived from recombinant or synthetic nucleic acid molecules into one or more human research participants. Note that the deliberate transfer of recombinant or synthetic nucleic acids into one human research participant conducted under a Food and Drug Administration or FDA regulated individual patient expanded access investigational new drug IND or protocol, including for emergency use, is not research subject to the NIH guidelines and thus does not need to be submitted to an IBC for review and approval. Also note that if an experiment falls into Sections 3A, 3B, or 3C, and one of the other sections, the rules pertaining to Sections 3A, 3B, or 3C shall be followed. If an experiment falls into Section 3F and into either Sections 3D or 3E, the experiment is still considered exempt from the NIH guidelines. Most non-exempt experiments that you may encounter in your research fall under Section 3D, experiments that require Institutional Biosafety Committee approval before initiation. Section 3D1 is where all experiments using Risk Group 2, Risk Group 3, Risk Group 4, or restricted agents as host vector systems. Section 3D2 are experiments in which DNA from Risk Group 2, Risk Group 3, Risk Group 4, or restricted agents are cloned into non-pathogenic prokaryotic or lower eukaryotic host vector systems. Section 3D3 are experiments involving the use of infectious DNA or RNA viruses or defective DNA or RNA viruses in the presence of a helper virus in tissue culture systems. Section 3D4 involves experiments that utilize whole animals for the experiments. See the links in our module for additional FAQs about animal experiments as well as the animal activities table from the NIH to better define where your animal experiments may fall. Section 3D5 involves experiments involving whole plants that require higher containment than BL1. Section 3D6 are experiments involving more than 10 liters of culture in any single vessel, and this would include all fermentation experiments. Section 3D7 are experiments involving influenza viruses. Section 3E are experiments that require IBC notice simultaneous with initiation. At UWM, they require biosafety review before initiation of the experiments and IBC review simultaneous with initiation. Section 3E1 are experiments involving the formation of recombinant or synthetic nucleic acid molecules containing no more than 2/3 of the genome of any eukaryotic virus. Section 3E2 are experiments involving whole plants. Section 3E3 are experiments involving transgenic rodents. The last section of Section 3 is Section 3F. These are your exempt experiments. At UWM, exempt experiments still require biosafety review prior to commencing the experiments, but do not require IBC review. Exempt experiments include the use of E. coli K12 derivatives to express genes from risk group 1 or 2 agents, recombinant DNA experiments involving Saccharomyces cerevisiae, propagating nucleic acids in a eukaryotic host that are originating from that species or similar, purchase, transfer, or use of rodents in BL1 containment that fall under any of the guidelines as outlined in Section 3F. For more information, visit the NIH guidelines site and review Section 3F for exempt experiments. There are additional sections to the NIH guidelines. These include Section 4 that outlines all of the detailed roles and responsibilities of personnel involved in NIH guidelines research. Section 5 provides footnotes and references from sections 1 through 4, and the appendices provide supplemental information referenced in the document, such as containment requirements, exempt experiments, previously NIH-approved Section 3A, 3B, and 3C experiments. If you need to report an incident, all significant incidents, violations, and research-related accidents and illnesses must be reported to the NIH Office of Science Policy within 30 days or immediately in the event of personnel exposures. For the reporting procedure at UWM, all incidents in BL1 and BL2 laboratories must be reported within 24 hours of the event occurring, if possible, using the online form link at uwm.edu/safety-health/first-report-of-biological-exposure-or-release-event. The Biosafety Program Office will follow up with you to finalize the report sent to the NIH Office of Science Policy and the Institutional Biosafety Committee. The PI is ultimately responsible for reporting the incident, but the Biosafety Program will submit the final report to the NIH Office of Science Policy. What should be reported? Any spills or accidents that result in potential exposure. Personal injuries involving recombinant or synthetic nucleic acid molecules, illnesses caused that are potentially associated with recombinant or synthetic nucleic acid research, a breach of containment associated with recombinant or synthetic nucleic acid molecules, in other words, a spill of BL2 or higher recombinant or synthetic nucleic acids outside of primary containment, improperly discarded recombinant or synthetic nucleic acid waste materials. Incident reports to the NIH Office of Science Policy should include the nature and consequences of the incident, the cause of the incident, and a detailed report of the response measures taken by the institution. As a reminder, there are additional resources available in this module on the Canvas page. Remember, noncompliance of the NIH guidelines does not just affect the persons in noncompliance. It affects all researchers conducting work subject to these guidelines and can impact NIH funding. When in doubt, contact your biosafety program with your questions about your work. This ends Module 7. Move on to Module 8 after completing the short quiz in Canvas.